Nucleic acids. Nucleic acids, whether DNA or RNA, are long polymers constructed from individual nucleotide units. These nucleotides are linked together by strong 3'5" phosphodiester bonds. This bond forms between the phosphate group attached to the 5' carbon of one sugar and the hydroxyl group on the 3 carbon of the adjacent sugar in the chain. This specific linkage gives each nucleic acid strand a distinct polarity. By convention, one end of the strand, the 5' end, often has a free phosphate group, while the other end, the 3' end, typically has a free hydroxyl group. The base sequence of a nucleic acid strand is always written by convention in the 5'3" direction, typically read from left to right. For example, a sequence might be written as 5' TCAG3 or simply TCAG. This convention is crucial for understanding the directionality of DNA replication, transcription, and translation. If the sequence is written in the reverse direction, the ends must be explicitly labeled, such as 3' GACT5. The positions of phosphates can also be indicated, for instance, PTPCPAPG, where P denotes a phosphate. In DNA sequences, a D may be included to specify deoxyribonucleotides, like DTDCDAG. In eukaryotic organisms, DNA is generally found as a double-stranded molecule, while RNA is typically single-stranded. However, exceptions exist, particularly in the viral world, where some viruses possess single-stranded DNA genomes, and others have double-stranded RNA genomes. Key characteristics of nucleic acids are that they are polymers of nucleotides linked by 3'5" phosphodiester bonds, they have distinct 3 and 5 ends, thus exhibiting polarity, and their sequence is always specified as 5'3". DNA structure. Double-stranded DNA exhibits several key structural features that are critical for its biological function and stability. Antiparallel strands. The two polynucleotide strands that make up the double helix run in opposite directions. One strand is oriented 5'3", while its complementary partner runs 3'5". This antiparallel arrangement is essential for DNA replication and transcription. Complementary base pairing. The bases on one strand specifically pair with bases on the other strand through hydrogen bonds, forming the rungs of the DNA ladder. Adenine always pairs with thymine, forming 2 hydrogen bonds. Guanine always pairs with cytosine, forming 3 hydrogen bonds. This specific pairing means that the base sequence on one strand precisely dictates the base sequence on its complementary partner, allowing for accurate replication and repair. Chargaff's rules. Due to this specific base pairing, the amount of adenine in a double-stranded DNA molecule is always equal to the amount of thymine, and the amount of guanine is always equal to the amount of cytosine. Consequently, the total amount of purines always equals the total amount of pyrimidines. These fundamental observations are known as Chargaff's rules. It's important to note that with a minor modification, these rules also apply to double-stranded RNA. Let's illustrate Chargaff's rules with an example. If a sample of double-stranded DNA contains 10% guanine, we can deduce the percentages of the other bases. According to Chargaff's rules, if guanine is 10%, then cytosine must also be 10%. This means G plus C totals 20%. Since the total percentage of all bases must be 100%, the remaining 80% must be composed of adenine and thymine. Because A equals T in double-stranded DNA, each must constitute 40% of the total. Therefore, if a DNA sample has 10% G, it will also have 40% T. This principle is extremely useful for determining unknown base compositions. Most DNA found in nature exists in a right-handed double helical form, famously known as Watson-Crick DNA or bDNA. In this structure, a hydrophilic sugar phosphate backbone of each strand is positioned on the exterior of the double helix, interacting with the aqueous cellular environment. The hydrogen-bonded base pairs, which are more hydrophobic, are stacked in the interior, forming the core of the molecule. There are approximately 10 base pairs per complete turn of the helix. The bDNA helix also features two distinct indentations along its surface, a wider major groove and a narrower minor groove. These grooves are important binding sites for various regulatory proteins that interact with specific DNA sequences without disrupting the double helix. A rarer, left-handed double helical form of DNA, known as zDNA, can occur in sequences that are particularly rich in GC base pairs. The precise biological function of zDNA is not yet fully understood, but it is hypothesized to play a role in gene regulation, perhaps by altering local DNA structure to influence protein binding. Supplementary point: bridge to pharmacology. Anti-tumor drugs targeting DNA. Several anti-tumor drugs exploit the unique structure and function of DNA to combat cancer by interfering with DNA replication or transcription. Doxorubicin and doxorubicin are examples of such agents, often called anthracyclines, used in treating leukemias and other cancers. They exert their effects by intercalating between the stacked bases of DNA. This intercalation causes unwinding and distortion of the DNA helix, which in turn interferes with the activity of topoisomerase II, an enzyme crucial for relieving DNA supercoiling during replication and transcription. By inhibiting topoisomerase II, these drugs prevent proper DNA replication and transcription, ultimately leading to cancer cell death. Other drugs, like cisplatin, a platinum-based chemotherapy agent used in bladder and lung tumor treatment, bind tightly to DNA, forming covalent adducts. These adducts cause significant structural distortion in the DNA helix. This distortion disrupts normal DNA processes, including replication and repair, leading to cellular malfunction and ultimately triggering apoptosis in rapidly dividing cancer cells. Denaturation and renaturation of DNA. The double helical structure of DNA is not entirely rigid. It can undergo reversible separation of its two strands, a process with significant biological and experimental implications. Denaturation. Double helical DNA can be denatured by conditions that disrupt the non-covalent interactions holding the two strands together, specifically the hydrogen bonds between base pairs and the stacking interactions between adjacent bases. This process results in the melting of the double helix, causing the two complementary strands to separate from each other. Importantly, no covalent bonds are broken during denaturation. Common denaturing agents include elevated temperatures, alkaline pH, and certain chemicals such as formamide and urea, which can disrupt hydrogen bonding networks. The temperature at which half of the DNA strands have denatured is called the melting temperature, which is higher for GC-rich DNA due to the three hydrogen bonds between G and C. Renaturation. If the denaturing conditions are slowly removed, the separated single-stranded DNA can renature or anneal. For instance, if a solution containing heat-denatured DNA is slowly cooled, the two complementary strands can re-establish their hydrogen bonds in base pairing, reforming the stable double helix. This process requires the strands to find their complementary partners and correctly align. This process of renaturation, or annealing of complementary DNA strands, is a fundamental principle in several important molecular biology techniques. For example, it is a crucial step in probing a southern blot, where a labeled DNA probe binds to specific target DNA sequences on a membrane, allowing for the detection of particular genes. It is also essential in performing the polymerase chain reaction, where short DNA primers anneal to template DNA strands to initiate DNA synthesis. When a well-characterized probe DNA binds to target DNA sequences that share sufficient complementarity, this process is specifically termed hybridization. Recall question: methotrexate affects which portion of the cell cycle? I. G1 phase, B. G2 phase, C. M phase, D. S phase. Answer explanation: the correct answer is D. S phase. Methotrexate is a chemotherapeutic agent that functions as a folate antagonist. It specifically inhibits the enzyme dihydrofolate reductase, which is essential for the synthesis of tetrahydrofolate. Tetrahydrofolate derivatives are crucial cofactors for the synthesis of purines and thymidylate, which are necessary building blocks for DNA synthesis. By blocking the production of these components, methotrexate specifically interferes with the S phase of the cell cycle, where DNA replication occurs, thereby preventing cell proliferation and targeting rapidly dividing cancer cells. Organization of DNA. Given the immense length of DNA molecules, especially in eukaryotes, they must be meticulously packaged within the confines of the cell's nucleus while still remaining accessible and functional for processes like replication, repair, and gene expression. This packaging involves multiple levels of condensation. Supercoiling. In certain biological contexts, such as mitochondrial DNA and the DNA of most prokaryotes, DNA exists as closed circular structures. These circular DNA molecules can exist in a relaxed state or in highly compact, three-dimensional structures known as supercoiled DNA. Supercoiling arises from topological strain on the molecule, caused by either underwinding or overwinding the double helix relative to its relaxed state. Imagine twisting a rubber band on itself; it will coil up to relieve the tension. Negatively supercoiled DNA. This form occurs if the DNA is wound more loosely than the standard Watson-Crick bDNA. This underwinding introduces torsional strain that is relieved by the DNA twisting upon itself in the opposite direction. Negative supercoiling is the predominant form found in cells and is essential for most biological reactions involving DNA, as it makes the strands easier to separate for processes like replication and transcription. Positively supercoiled DNA. This form occurs if the DNA is wound more tightly than in Watson-Crick DNA. This overwinding also introduces strain. While less common, positive supercoiling can occur transiently ahead of a moving replication or transcription complex. Topoisomerases are a class of essential enzymes that are responsible for changing the amount of supercoiling in DNA molecules. They achieve this by making transient breaks in the DNA strands, either one strand or both strands, alternately breaking and resealing the sugar phosphate backbone. This allows the DNA strands to unwind or rewind, thereby altering the supercoiling level. For example, in the bacterium Escherichia coli, an enzyme called DNA gyrase specifically introduces negative supercoiling into DNA, facilitating its replication and transcription. Topoisomerases are also targets for some antibiotics and anti-cancer drugs. Nucleosomes and chromatin. In eukaryotic cells, the nuclear DNA is found in a highly organized complex called chromatin. Chromatin is composed of DNA associated with a variety of proteins, primarily histones and various non-histone proteins. The fundamental packaging unit of chromatin is the nucleosome. Histones are a family of small, highly conserved proteins that are exceptionally rich in the basic amino acids lysine and arginine. These amino acids confer a strong positive charge on histones, which is crucial for their ability to bind tightly to the negatively charged phosphate backbone of DNA. There are 5 main types of histones: H1, H2A, H2B, H3, and H4. The core of a nucleosome is formed by an histone octamer, which consists of 2 copies each of 4 histone proteins, H2A, H2B, H3, and H4. These 4 histones are highly conserved across eukaryotes. Approximately 147 base pairs of DNA are wound almost twice around the outside of this histone octamer, forming a single nucleosome core particle. A series of these nucleosomes connected by stretches of linker DNA gives chromatin the characteristic appearance of beads on a string under an electron microscope. And this structure is more formally referred to as a 10 nanometers chromatin fiber. This level of packaging reduces the length of DNA by about 7-fold. A 5th histone protein, histone H1, is associated with the linker DNA located between nucleosomes, often at the point where DNA enters and exits the nucleosome core. H1 plays a crucial role in further compacting the 10 nanometers fiber. It helps to pull the nucleosomes closer together, causing them to coil into a more condensed, solenoid-like structure known as a 30 nanometers fiber. This provides an additional 6-fold compaction. This 30 nanometers fiber undergoes even further levels of condensation, forming large loops that are attached to a protein scaffold, and then coiling and folding to eventually form the highly compact structure of a visible chromosome. Each eukaryotic chromosome in the G0 or G1 phase of the cell cycle typically contains one linear molecule of double-stranded DNA. Within the interphase nucleus, there are 2 primary types of chromatin, distinguished by their condensation state and transcriptional activity. Euchromatin: this is the more loosely packed and decondensed form of chromatin. It generally corresponds to the 10 nanometers nucleosome fibers and loosely associated loop 30 nanometers fibers. Euchromatin is considered transcriptionally active or potentially active, meaning its genes are more readily available for expression by RNA polymerase and other transcription factors. It stains lightly with DNA binding dyes. Heterochromatin: this is a much more highly condensed and tightly packed form of chromatin. It is typically associated with regions of the chromosomes that are transcriptionally inactive or silenced. There are 2 main types: constitutive heterochromatin, always condensed, found in regions like centromeres and telomeres, and contains highly repetitive DNA sequences, and facultative heterochromatin. Heterochromatin can be observed as dense, dark staining regions in the interphase nucleus. The packaging of DNA in eukaryotic cells thus progresses from a double helix to 10 nanometers chromatin, then to 30 nanometers chromatin, and finally to higher order packaging where the 30 nanometers fiber forms loops attached to scaffolding proteins, resulting in the visible chromosome. The nucleolus is another prominent structure within the interphase nucleus. It is a specialized, non-membrane-bound nuclear region primarily responsible for the synthesis of ribosomal RNA and the assembly of ribosomes, which are essential for protein synthesis. It is often surrounded by heterochromatin. During mitosis, the entire DNA content of the cell becomes extraordinarily highly condensed. This extreme compaction is essential to allow for the orderly separation of the sister chromatids into daughter cells without tangling or breakage. This mitotic phase is the only time in the cell cycle when individual chromosome structures are clearly visible under a light microscope. Chromosome abnormalities, such as changes in number, translocations, deletions, inversions, and duplications, can be assessed on these mitotic chromosomes using techniques like karyotype analysis, typically on metaphase chromosomes, where they are most condensed, and banding techniques.